So, welcome to my talk today. This, indeed, as Frederick said, it's the title of my PhD thesis, which I was working on for the last too many years, like 7, I think. I did it here at the Hohenheim University, which is an agricultural university in the southwest of Germany. And yeah, why did I choose this topic? So, yes, here, no, so fast. Okay, so here's the issue. I don't know if you all are aware why phosphorus is an issue for world food security. Yeah, phosphorus is one of the micronutrients and very important for the growth of agricultural crops for food, fuel, and timber and commodities. So, but the problem is that phosphorus fertilizers have a very low efficiency. This is because phosphorus in its main present form in the environment is in the form of phosphate. Yeah? And phosphate interacts strongly with anything it finds. So soil particles and soil organic matter and calcium and on iron, on aluminium, hydroxide. So when you throw a drop of phosphate on soil, the soil takes it and doesn't let it go. So we have the situation that we have a lot of phosphorus in our soils, but with a very limited availability. Okay. So we have this low efficiency of phosphorus fertilizers. And then we can say, yeah, we have fertilizers, we can mine them, we have mineral deposits, but they are limited, it's a limited resource. And additional, there's a geopolitical issue because 90% of the world's phosphorus reserves are in Western Sahara, which is occupied by Morocco. So there is another topic of conflict. And phosphorus fertilizer prices have a big impact on food prices. So that's a problem. So we, until now, as we all know, as was happening also with other fertilizers or other nutrients, that the organic nutrient was not really investigated, because usually fertilizer industry was interested in chemical fertilizers because they are more easy to calculate, more easy to apply. So this means that we have some knowledge about inorganic phosphorus, but very limited knowledge about organic P. So a lot of the phosphorus in the soils outside is in the forms of organic P. And this organic P is a resource we could use with a good management of our agricultural fields. So this organic P can be accessed by, or the availability of the phosphorus in these pools is controlled by plant-soil-microbe interactions. So microbes control the door, they have the key to organic P. And plants feed microbes. So if we use plants wisely, we can manage these key pools. Therefore, our suggestion is that cover crops, which is green manures, that they could be a promising tool for phosphorus management. Here we have a picture where you have one crop with cover crops and the other one without. And there you can imagine what how the microbial community, that probably there will be differences between the two fields. So we had specific goals, which was how to investigate the mechanisms how cover crops increase the access to phosphorus for land crops, and looking for the role of soil microbes, as we are soil microbiologists. And we wanted to look at, this is now more basic science, the soil microbial functions and how they are connected to enzymatic availability of these phosphorus pools. And then more on an agronomic, from an agronomic perspective, how management, like cover crop choice, because you can imagine that the sunflower is not the same as a clover, and tillage, because if you move around the soil, the microbial community changes and the physical-chemical conditions change. If this and how this affects the plant-soil-microbe interactions. Yes. So for this, we conducted 3 studies. First of all, a large meta-analysis. Okay, large. Maybe there are larger meta-analysis, but for me it was a large meta-analysis. Where we took studies from all over the world, because there is not so much about phosphorus and green manures. So we took these studies, and from these studies we developed a conceptual model how these cover crops change the peat benefit. And to test these hypotheses, we performed 2 field experiments in southwest Germany, always with winter cover crops before soybean. Soybean was the main crop, in loess-derived soils. Yeah, this is important because loess is maybe fine particle. It's very fine particulated, so there's very little sand, and this has effects on the soil microbial community. Yeah, and we assessed the organic and inorganic peat pools and microbial properties. So study number 1. In study number 1, we had different cover crop species in pure culture, and we sampled the rhizosphere of them by taking samples from the rhizosheet, which is the soil that is adhering to the roots. In study number 3, we had more of a cover crop mixture and we took direct bulk soil samples, but there we were more interested in the interaction of cover crops and different tillage treatments. So here again, study number 1, which was the meta-analysis, where we looked with these keywords, gathered a bunch of articles, and then screened them by different characteristics. And so then we could assess how much biomass was produced by cover crops, how was their peak concentration, which effect had the cover crops on the nature performance, if you had a yield increase or phosphorus concentration. And we looked for the few studies, there were only few studies which analyzed the mycorrhiza, for example, or microbial P or enzyme activity. And we look for management interactions, and if soil P availability has an effect, because you can imagine that it makes a large difference if you have if you work in a soil with very little available phosphorus, or if you work in a soil which is heavily over-fertilized, as most of soils in Catalonia, for example, because of manure applications. Yeah. I'm a big fan of R, so we use a lot of R, and yeah, you will see the effect. Then we had slide number 2, which is a press, accepted and yeah, press. So here we had a field experiment, as I said, with these 3 cover crop species, which have quite different properties regarding their relation with mycorrhiza. And also, for example, mustard is known to produce a very large biomass, but buckwheat in this region not so much. And Phacelia is known to interact a lot with the soil microbial community. Okay, so here what was the interesting thing was that we had a cover crop phase, and then in spring a short fallow phase, and then again the soybean phase. And we had these different sampling points. And when we could, we sampled also rhizosphere. So here we sampled the rhizosphere of the cover crops, and here we sampled the rhizosphere of the soybean. And we had our study number 3, where at the research farm, we had a full tutorial experiment with 2 tillage treatments, like direct seeding or no-till, and mulch seeding, which is a kind of reduced tillage, and 2 cover crop treatments, the spare fallow and cover crops, in 3 replicates. And so here again, we something what happened to us is that we had produced too much data, and then we could use only a very little amount of this data. So we had all these sampling dates and analyzed everything, but then I wrote the publication only with these 2 data points. So when you do something, don't produce too much data. You know? Yeah. Let me tell you some words about the methods we used, because I'm not sure if you are here in aquatic microbiology or specifically here at this institute, if you use the same methods as we in the soil science did. So we measured enzyme activities of different phosphorus cycling enzymes. We measured microbial biomass P. We looked for the microbial community structure, but not with fancy metagenomics, but with old-fashioned PLFAs. Here we did a little bit about 16S, and specifically for D, which is a functional gene coding for alkaline phosphomonoesterase. So there we had an interesting relation. We could draw a relation between the gene and its expression. And yeah, and in study number 3, we did also some. We added, for example, some sugar to soil samples, and then measured the produced CO2. So to assess the microbial biomass, the microbial respiration, and therefore the biomass. And we did this with different amounts of nutrients to assess nutrient limitation. And we did biology in cooperation with Magdalena Prak from Poland. So, just at a glance, very briefly, how we did the enzyme activities. We have these wonderful substrates you buy from your chemical supplier, and this is here. You have the substrate which is interesting for you, and your enzyme in the soil wants to cut this here. And here, this one is a fluorescent part of the molecule. So if you incubate, if you add to your soil this substrate and incubate it, your enzymes will cut here and it will start to glow. And then you can measure it in a fluorescence reader. So you have a quantification of how fast the enzymatic reaction is performed in the soil, and you can quantify it. The PNIC, the phosphorus in microbial biomass, we did hexanol fumigation extraction, where you take a soil sample, add hexanol, shake it a lot, have there inside anion exchange resins, which absorb the released phosphate, because the cell walls, of course, they get destructed. And then you have your phosphate on these little anion resins, and where you can release it afterwards and color it and measure it photometrically, right. And the third one, which was quite important for us, was the PLFAs. As you all know, cell membranes are made of phospholipids or nucleolipids. So the idea is that you extract them with heavy solvents. Then you can identify them in a gas chromatograph and compare them with a standard mixture. And we have the luck that there are specific marker biomarkers. Yeah, and with these, with the fatty acid analysis, we can, for example, we can separate gram-positives, gram-negatives, and cytotrophic fungi and agroscular mycorrhizal fungi. Yeah, as I said, it's not metagenomics, but it gives you an interesting idea of what is there. That was the easy part. We did also one very interesting new method, which was developed by some colleagues in Zurich, which is because, until now I always talked about soil organic P, but what P? Yeah, there are so many different kinds of molecules which can be organic. You can have monoesters, you can have diesters, you can have weird phytates, for example. Phytate is a storage. It's a complicated ring and molecule where you have up to 6 phosphates on a carbon ring, and which is one of the most important organic P compounds in the outside. So when we did not want to talk only about soil organic P, but we wanted to characterize, to have a look into this black box. So we used this enzyme addition assay, because before I explained to you how we measure enzyme activity, but here in this case, we added enzymes, yeah? In this case, so we extracted the organic P from the soil, and then we added one of these enzymes or combinations, and this brings you to these wonderful colored plates, which are very complicated to work with. And then you can quantify the released phosphate. So, because then we can look, for example, if the phytase releases a larger amount of phosphate than the nuclease, for example, then we know that we have more phytate in our soil than phosphodiesterase. Yeah, and this is a very interesting approach. So we know not only the process, which is what we would do in the enzyme activity measurements, but we can have a look at the pools. Okay, this is what comes out from this enzyme available from this enzyme addition assay. Here on the left, we have different P pools. Here, the white one is the inorganic P, and these two red ones are the organic P. This is enzyme stable, this is enzyme labor. Here we see already that organic P is an important proportion of soil P pools. But standard soil tests, which are performed in agriculture, only have a look at these pools, and they completely forget this phosphorus. And here, the enzyme addition assay allowed us to characterize even further this enzyme labile pool. So here we see in the different colors, we have deasterase labile organic P, monosterase labile organic P, and phytase labile organic P. And here what you could see is that in this case, in the treatment with cover crops and not inversion tillage, that had the highest amount of enzyme labile organic P. This means that the cover crops in this treatment were able to increase the amount of organic P which is available for degradation. Yeah. This one now is from the other experiment where we assessed the rhizosphere of single cover crop species, where we took out the cover crop and with a toothbrush we took the samples of the rhizosheet. So, and here what we see also, you see, first of all, you see it is a different field. In this field, the inorganic P is much less than the organic P. So you see already that there's a very large heterogeneity out there from field to field. It depends very much on your soil and your agricultural management. But here it's even, here the difference is even greater. If a farmer would only look at this phosphorus pool, then he would have a very wrong picture of reality. And here what we saw is that in the cover crop soils, that the amount of enzyme labile P was also increased, mainly because of increases in deasterase labile P and monosterase labile P. This means that near the root of cover crops, we have more free diesters and free monoesters which can be attacked by enzymes. Yes, I wanted to say. So, now coming back to agronomy, we wanted to, we had a look if how this was a figure from the meta-analysis, and here we see the effects of different cover crops. We aggregated them to families, yeah, to cover crop families like lupinus, brassicacea, serraceae, fabacea. And this one is the main crop. So here we see already a large difference between different main crops, for example, because we see that cereals were rather unimpressed if you were growing a green manure before, but maize was highly responsive. This is very interesting and explains something about the variation, because in some experiments there are positive results of cover crops, and in others there's no effect, or yeah. So here we have this large thing. So we see that maize is most suited for cover cropping. But also we have even an unfavorable combination. For example, if you grow Quassia before cereals, then you have a negative yield effect, which makes agronomically perfect sense, because the cereals are members of the Quassia, and you should not grow always the same plant on the same fields. Yeah. But here, what we could say is that main crops, they differ in their ability to take advantage of cover crops. Then we have to look how much phosphorus was cycled through the cover crop biomass. We have seen here that on average, there was around 7 kilograms of phosphorus cycling per hectare cycling through cover crop biomass. This means in 1 hectare, your cover crops take up 7 kilograms of phosphorus and store it in their biomass. And when you cut them down afterwards, these 7 kilograms of phosphorus, they get degraded and mineralized and are again in the soil. Yeah, this is just the cycling through cover crop biomass. We have an interesting case. This is not so much microbiological, but it's important to understand the picture and the mechanisms and processes. Here we have again an interesting thing, which is that again the Quassians, they have the largest biomass and therefore a very large P content, but they have a very small P concentration. Yeah, you can imagine that like cereals or grasses, that they produce a lot of biomass, but like very good biomass. And this could be an explanation why this large amount of phosphorus was not able to give to increase yields, for example, here, because it has a very low C to P ratio, yeah, and therefore it cannot be quickly degraded by the microbes. So now again in the microbial world, where we feel best. Here we have, this is also from meta-analysis, we have the percentage change with cover crops of mycorrhizal abundance and of microbial P. So what we see is that these are the mycorrhizal cover crops, and this is the control. So we see that mycorrhizal cover crops increase AMF abundance greatly, which makes perfectly sense, but here we could find that. And we also saw that the pH pool of microbial phosphorus, that this was also increased by quite a lot, about 25%. And if we think about that, the phosphorus stored in the microbial biomass, as we have a lot of microbial biomass in soils, we have a lot of phosphorus inside this microbial biomass. So we have between 5 and 70 kilograms on average, or a range, in field soils. So increasing this by 25% is a lot of phosphorus. And phosphorus that is stored in the microbial biomass is moderately available for plants, because microbes, they die or things like this. No? Okay, so we have seen that cover crops, they interact with the microbial community and increase abundance of the P storage, and also some specific groups, because, for example, mycorrhiza are very important if they interact strongly with the plants and interfere in the P uptake. Is this everything too basic, or is this okay like this? Okay, wonderful. Okay, so this one is here we have seen also we have a figure where you can see here these the two brown treatments are the bare fellow, green is the cover crop treatment. This is from study number 3. And we see that cover crops clearly increased the microbial abundance. And here we measured as PLFAs. As a first summary, we can say that the cover crop families, this is a result of a meta-analysis, and we can see that the cover crop families like Rassicacea, Fabaceae, and Leucaena and Poaceae, there we have most of the data, they have distinct properties. Because here on this radar chart, we have on these axes, we have different properties. Like here, for example, is how much biomass the cover crop produces, how much P content to P concentration, which is the effect on mycorrhizal abundance, on microbial P, phosphatase activity, and available phosphorus as inorganic phosphate. And here, the more on the outside the point is, the line is, the greater the greater. Yeah, it's a relative comparison. And here we see, for example, that the Poaceae, that they produce a large amount of biomass with the lowest P concentration, but they have also a good effect on mycorrhiza and microbial phosphorus, not so much on phosphatase, and they decreased the availability of phosphate in the soil because of their high CP ratio. Because if you add a plant material with a large amount of carbon inside, then your microbes, they will take the nutrients from, they will immobilize nutrients from soil, no? And on the other hand, for example, we have, I don't want to show you all of the, explain every single family, but you see that we have large differences among the families. So again, when talking about cover crops, it's important which species is used. Then, the other thing, the species which we use forms part of the agronomic management of the crop rotation. And another important thing of agronomic management is tillage. So here we have seen in this experiment, we have seen that the fungal abundance measured as PLFA that was strongly increased in the cover crops treatment with reduced tillage. Here the most, also there was quite a variability, but here we had this interaction. And so we see that we saw a similar thing for gram-negatives, and therefore we can say that that agricultural management hasn't interacts with the cover crops. So we have to think this is unfortunate because it makes science a little bit more difficult because we cannot aggregate all the different experiments, but it is very good because this means that farmers have the possibility to adapt their management to their needs. And the other thing, what we found, is that in this one is study number 2, again here the players are the fungi, that we had a massive increase of fungal PLFAs around the roots of cover crops. So therefore, we want to stress that saprotrophic fungi probably play an important role in the cover crop effect, and in as I showed in the beginning, these the changes in availability of organic peat pools. So the role of saprotrophic fungi, which sadly don't exist so many primers for them, they are very often overlooked. So please look at fungi. They're important. And we, as I told you in the beginning, we also looked at the functional gene 4D. This one, so 4D produces an alkaline, a bacterial alkaline phosphatase. And here, what we wanted to look is, we have always this, or at least me, I can speak for you. we have always this conception that plants interact with the soil and the soil microbial community, and they specifically will feed microbes that give the benefits. Would make sense. No? If I'm a plant, I feed the microbes that give me benefits, and I kill the microbes that do something bad to me. So I expected that if we measure 4D in the rhizosphere near plants, that I would find or an increased abundance of 4D, because the plant, as this 4D codes for phosphatases, the plant would have a benefit from a large amount of 4D, and the plant would also have a benefit from a large of phosphomonoesterase activity. But in those cases, there was no difference between bulk soil and rhizosphere soil. So we found no evidence that the plant controls what is happening in the rhizosphere. In this experiment, at least, it looked like that plants feed the microbial community, but they have not the ability to tell the microbe, please produce more 4D, or please express your 4D to produce enzyme activity. So the acceleration of P cycling, we expect it to be caused by a general increase of microbial abundance, because we have seen a lot of evidence that there was a massive increase of microbial abundance, but not so much that the plants control who is going there. Okay, now wrapping up the things I found. Here we have 3 different pathways of key benefit. Yeah, I will explain it in detail. Which is, first of all, we have a plant biomass pathway. Here we have the cover crop is growing, then here comes winter, and then the cover crop is killed and is there as a residue. Okay, so what happens is you have a large amount of phosphorus, of inorganic and organic P in the soil. This inorganic and organic P is taken up by the cover crop. There is some interaction with the microbe, but in this case, in the plant biomass pathway, this, for example, plant species that grow very large, like brassica, like rapeseed or mustard, or things like this, they take up a lot of P from soil, store it in their biomass with a very large biomass. Then they get cut down. And then you have here the residues, and these residues, they get mineralized by the microbial community here, and are made available for your main crop. This is one of the modes of action of cover crops. Here, its advantage is if you have a low CP ratio, because if you have a lot of carbon in your plant biomass, then here the phosphorus here will be immobilized and won't be made available for the main crop. And your decomposition dynamics matter, because then you depend on climate, for example, because if it's very dry, then your microbes won't work and these residues don't get decomposed. We have the microbial enhancement pathway. This one is the most interesting one for me. Here you have microbes. You have a cover crop which feeds microbes by root exudates, for example. And this enhanced microbial community is able to take up phosphorus from different sources of the soil, from different pools of the soil, and is able to store phosphorus in its biomass, but also has special abilities, because later, when this cover crop doesn't need to grow so big and to transport so much phosphorus in its biomass, because it left a legacy of an enhanced microbial community in the soil. And then when your main crop grows, your main crop becomes delivered by your microbial community with phosphorus. Imagine, for example, if you have mycorrhiza in soil, And this is a cover crop that increases the amount of mycorrhiza spores in soil. Yeah. Then, if you have a lot of mycorrhiza spores, then this main crop finds immediately mycorrhiza spores and it gets colonized. So it has a greater mineralization. So here, crop rotation gets very important because mycorrhiza, not all crops are mycorrhiza. Thank you very much. The third pathway is the biochemical modification pathway, where we have some specific cover crops, for example, members of the Lupinus genus, that have very special abilities with their roots to produce carboxylates, to decrease soil pH. And so they change, they modify the conditions, the biochemical conditions of the soil, and so they release, they are able to access soil pH pools nobody else is able to. And so these, if they improve the soil pH availability, this is also a benefit for the main crop. So these were the three pathways of P benefit. And it's not so easy because, as always, these three pathways, they take place simultaneously. But we could confirm that the cover crop biomass has a very large potential because it can cover a significant amount of the phosphorus that is needed by main crop, can be covered from the phosphorus stored in the cover crop biomass. And we could see that we have increases in the availability of soil organic P, and that this is related to microbial abundance and microbial activity. So we identified a strong role of microbes in these processes. And we found these interactions of cover cropping and tillage and the effect of cover crop species. So we state that the P benefit that it can be optimized by management decisions. So my point is that cover crops are awesome, they are multifunctional, and this is an additional thing. Cover crops finally can help us to bring agriculture to an agricultural system based on ecosystem functions. Because in the last 60 years or so, most of agricultural science intended to make agriculture more reductionist, let's say like this. Yeah. So you have simple systems. You don't have different crops. You kill all weeds. You don't even think about organic nutrient pools in the soil because everything you need, you bring it with a bag from your supplier. And if farmers, if they are convinced to use cover crops because they bring them benefits, then they are using. Then they are again they are looking at ecosystem functions. And so my hope is that when farmers do this, they will start also to see many other benefits like of biodiversity, for example. And that this slowly might change our agricultural system. But as always, we end up with more open questions than we could answer at the beginning. So there are the cover crop species. There are many differences. So we could look to, because as you know, there are many thousands of different plant species. We can test all the plant species, but what we can do is to aggregate them by functional traits. Yeah, the plant functional traits. This is like root biomass or things like this. So you put together quite different families or species of plants, but according to their traits. This is a very functional approach. We have to look here as an enter, so we have to look at the rhizosphere extension, because as we have seen, there's a lot of processes are happening in the rhizosphere, and in the rhizosphere is a hotspot of microbial activity. So we have to know how large is our rhizosphere, because if we have our plants there, and we know that our plant do a lot in its rhizosphere, we need to know the volume and extension of the rhizosphere to be able to make predictions. We need to understand more about agronomic management and site conditions. And the only way to do that is to do participatory approaches in collaboration with farmers, because the farmers know what's relevant to them. So, yeah, these are more the general remarks, and the very specific questions, the more specific questions would be, what is the origin and the fate of the mineralizable organic peat pools? Because we found, yeah, there's an increase of organic peat pools with cover crops, of enzyme available organic peat with cover crops. But what is the from the perspective of microbial ecology, where does it come from and where does it go? We want to know more about the role of saprotrophic fungi. And here, what I said before, what we saw with the 4D expression, do plants have control over what happens in their rhizosphere, or do they just pump root exudates in soil, hoping and praying so that the right microbes grow there that can support them? So, yeah, and a better knowledge on this point will enable us to understand agricultural management effects on biodiversity-related functions, because as all these things are related to microbes, yeah, because we talk a lot about biodiversity, but when it gets to specific functions, we don't really understand what's happening. So, thank you for your attention. And we have many very kind collaborators and fantastic team at Hohenheim, and Hans Lammers, and also Hans Lammers is from Australia, but he supported me a lot. Yeah, so thank you very much. Any questions?
I have a question.
Yes.
Well, first of all, thanks for your presentation. I like very much, but I need some time to process all the information. But you said that the plants exudate some materials to feed, mainly to feed the microbials, the soil microbial. But there are some plants that also exudate some organic acids.
Yes.
That I suppose that will increase the soil pH, it will come to the ability of phosphorus.
Yes.
So will be a direct effect of plant on the phosphorus ability. So do you have considered this to study also the organic acid oxidation from plants?
Yes, we studied this was in another, I participated in a study where they measured carboxylates, the root exudation of carboxylates. The problem is that in the field, it's nearly impossible to assess this, because because there are like methodological problems to really see how much how much carboxylates are exuding from the plants. But yeah, the carboxylates, they represent a significant amount of the carbon exudation and carbon deposition into soil. Yeah, and additionally, they have these physical chemical effect on soil and on aggregation, on pH, as you said. And they are used as feedstuff by different soil microbes and different communities. So, as I said at the end, the third pathway, which was more of kilos, for example, their carboxylates are heavily involved in this pathway. So the plant root exudes carboxylates, and these carboxylates shape the phosphorus availability in the soil. Also, other plants exude carboxylates, but they're like very prominent in the location.
Okay, thanks.